Welcome to BBIU. This episode is based on a public article from Biopharma Business Intelligence Unit, an independent strategic intelligence platform analyzing biopharma, regulation, geopolitics, capital, and structural risk. We do not treat events as isolated headlines. We read them as signals inside larger systems. This podcast provides a concise public entry point into the issue, while the institutional version develops the full structural assessment for decision makers facing high consequence uncertainty. FDA's draft guidance and the repricing of nonclinical evidence in oncology, May 31st. From animal study volume to human relevant evidence architecture, institutional relevance snapshot. What happened? In May 2026, FDA issued draft guidance on streamlined nonclinical safety studies for certain oncology biologics and conjugated products. The guidance focuses primarily on 3-month general toxicology studies and recommends ways to reduce unnecessary animal use while maintaining patient safety. FDA emphasizes pharmacologically relevant species, weight of evidence risk assessment, and fit-for-purpose new approach methodologies when appropriate. Why this matters now? The guidance does not broadly eliminate animal testing in oncology. Its importance lies in a narrower but more consequential shift. FDA is making certain animal studies conditional on biological relevance, incremental safety value, and the strength of the supporting evidence package. This changes how sponsors, CROs, regulatory affairs teams, and investors should evaluate non-clinical development strategy. Who should care? This issue is relevant for oncology biotech sponsors, regulatory affairs teams, preclinical CROs, translational science groups, clinical development leadership, business development teams, investors, and capital allocators evaluating oncology platforms. What kind of decision this affects? The guidance affects IND-enabling strategy, CRO selection, pre-IND planning, non-clinical budget allocation, Module 4 preparation, Module 2.4 narrative construction, and investor assessment of preclinical readiness. Executive summary. The visible event is FDA's draft guidance on reducing unnecessary animal testing for selected oncology products. The deeper issue is the repricing of non-clinical evidence. This guidance should not be read as deregulation. It does not remove the sponsor's responsibility to protect patients. It does not make animal testing irrelevant. It changes the standard by which certain animal studies are valued. The key shift is from animal study volume toward biological relevance. A study that is technically compliant but biologically weak may no longer carry the same regulatory value. A more selective package may be acceptable if supported by relevant pharmacology, target biology, prior evidence, weight of evidence reasoning, and appropriate alternative methods. For sponsors, the question is no longer only whether the expected animal studies were performed. The question is whether the selected evidentiary system is appropriate for the biological risk being assessed. For CROs, the future value proposition may shift from study execution toward evidence interpretation. The strongest providers will not simply be those able to run more animal studies, but those able to help sponsors justify which studies are necessary, which are redundant, and which alternatives are regulatorily defensible. Observable surface, FDA's draft guidance is titled Oncology Pharmaceuticals, Streamlined Non-Clinical Safety Studies for Biologics and Conjugated Products. The document is draft guidance and is distributed for comment purposes. FDA states that, when finalized, the guidance will represent the agency's current thinking, but it does not establish legally enforceable responsibilities. The purpose is to assist sponsors in implementing streamlined approaches for non-clinical safety assessments of certain oncology pharmaceuticals. FDA states that the guidance is intended to facilitate development of biologics and conjugated products for cancer treatment while avoiding unnecessary animal use. The document focuses primarily on 3-month toxicology studies for certain oncology pharmaceuticals and supplements ICH-S9, ICH-S9 questions and answers, and FDA guidance on oncology therapeutic radiopharmaceuticals. FDA's recommendations include several specific pathways. For biologics, animal toxicology studies should use pharmacologically relevant species, supported by pharmacology studies demonstrating target binding and intended pharmacologic effects. If no pharmacologically relevant species exists, safety assessment may be based on a weight of evidence risk assessment instead of animal toxicology studies. For biologics with activity similar to humans in both rodent and non-rodent species, general toxicology studies may be conducted in a single rodent species and supplemented with weight of evidence assessment when appropriate. For PD-L1 blocking monospecific antibodies, chronic effect assessment may be based on weight of evidence instead of a 3-month toxicology study. For CD3 bispecific T-cell engagers, chronic effect assessment may also be based on weight of evidence instead of a 3-month toxicology study. For ADCs with cytotoxic payloads, where payload safety is well characterized and the payload is the main toxicity driver, a 3-month study may be conducted in rodents only under specified conditions. FDA also states that a weight of evidence assessment may include investigational product data, literature-based target assessment, toxicity findings from related products or classes, and other appropriate data, including fit-for-purpose NAMs. What the surface does not explain. The guidance explains which animal studies may be streamlined under certain conditions. It does not fully explain how the burden of regulatory reasoning shifts inside the sponsor organization. It does not directly address CRO business models. It does not tell investors how to distinguish between a sponsor that is intelligently streamlining nonclinical development and a sponsor that is simply underbuilding its safety package. It does not resolve the central operational problem. If fewer animal studies are performed, the remaining evidence must become more biologically relevant, better integrated, and more defensible. That is the real institutional issue. The guidance does not reduce the need for safety evidence. It changes the form in which safety evidence must be justified. Structural diagnosis FDA is not simply reducing animal testing. FDA is changing the value structure of nonclinical evidence. Under the older model, regulatory confidence could often be supported by familiar study packages, animal toxicology, repeat dose studies, safety pharmacology, toxicokinetics, and standardized GLP execution. Under the emerging model, the familiar package is no longer sufficient by itself. A study must answer a biological question. A species must be pharmacologically relevant. A 3-month study must add meaningful safety value. A NAM must be fit for purpose. A weight of evidence assessment must be interpretable. The system being reshaped is the evidence production system behind oncology drug development. The burden being transferred is not patient safety responsibility. That remains with the sponsor. The burden being transferred is the burden of justification. Sponsors must now defend not only the studies they perform, but also the studies they omit, shorten, replace, or streamline. Force breakdown regulatory force. FDA is using guidance to move toward more selective non-clinical safety assessment for certain oncology biologics and conjugated products. The agency is not eliminating non-clinical review. It is requiring better alignment between study design and biological relevance. Scientific force. Targeted oncology products do not always behave in animal species in ways that meaningfully predict human risk. Biologics, ADCs, bispecific antibodies, and T-cell engagers can raise species relevance problems that make conventional animal models incomplete or misleading. Economic force. If some animal studies lose regulatory value, the economics of preclinical outsourcing may shift. CROs dependent on animal study volume may face pressure. CROs able to support human-relevant models, translational pharmacology, computational toxicology, and regulatory evidence integration may gain strategic value. Operational force. Sponsors must integrate pharmacology, toxicology exposure, product class knowledge, NAMs, CMC comparability, clinical planning, and regulatory strategy into a coherent package. Strategic force. The competitive advantage moves from performing more studies to constructing better evidence. What is most likely being underestimated? The first underestimated issue is the burden on regulatory affairs. This guidance makes Module 4 more strategic, but it also increases the importance of Module 2.4. Module 4 contains the non-clinical evidence. Module 2.4 must explain why that evidence is sufficient. The second underestimated issue is CRO selection. A CRO that can execute animal studies is not necessarily the CRO best positioned for this environment. Sponsors will need partners that can help justify model relevance, interpret alternative evidence, and support regulatory dialogue. The third underestimated issue is investor misreading. A reduced animal study package can mean two different things. It may reflect scientific maturity and regulatory alignment, or it may reflect an underdeveloped safety strategy disguised as efficiency. The fourth underestimated issue is NAM overstatement. NAMs are not automatically superior because they are non-animal. In the FDA guidance, they are part of a broader weight of evidence framework. Their value depends on purpose, validation, interpretability, and regulatory context. Forward scenarios. Scenario 1. Discipline streamlining trigger. Sponsors engage FDA early and justify streamlined nonclinical packages with strong pharmacology, target biology, prior evidence, and weight of evidence reasoning. What it would look like: more single-species approaches, fewer redundant 3-month studies, selective use of NAMs, stronger Module 2.4 narratives, and more efficient IND-enabling timelines. Institutional consequence. Strong sponsors reduce cost and time without weakening regulatory credibility. Scenario 2. Superficial cost reduction trigger. Sponsors interpret the guidance mainly as permission to cut animal studies. What it would look like: thin scientific justification, fragmented NAM use, weak species relevance arguments, FDA questions, additional study requests, or delayed IND progression. Institutional consequence. Apparent efficiency becomes regulatory exposure. Scenario 3. CRO market repositioning. Trigger. Sponsors begin selecting CROs based not only on GLP capacity, but on evidence integration capability. What it would look like: increased demand for translational toxicology, fit-for-purpose NAMs, computational modeling, regulatory strategy support, and integrated Module 4, Module 2.4 preparation. Institutional consequence. CRO value shifts from study execution toward evidence architecture. Institutional exposure. Institutions are exposed if they interpret the guidance as a simple reduction in animal testing. Regulatory affairs teams may underestimate the need to defend omitted studies. Executive leadership may treat streamlined non-clinical development as a cost-saving opportunity without recognizing the increased burden of justification. Investors may misread a smaller animal study package as either automatically efficient or automatically risky, instead of asking whether it is biologically and regulatorily defensible. CROs may overestimate the durability of animal study volume if they fail to develop interpretive and human-relevant evidence capabilities. Business development teams may misjudge platform maturity if they do not evaluate the quality of the non-clinical evidence architecture behind the asset. The most dangerous lag is not technical, it is interpretive. Organizations that continue to evaluate non-clinical development by study volume alone may misprice regulatory risk. Why this matters? This matters because non-clinical evidence is one of the earliest points where clinical, regulatory, economic, and ethical risk converge. A weak non-clinical package can delay development, trigger regulatory questions, undermine investor confidence, and expose patients to poorly characterized risk. A well-constructed, streamlined package can reduce unnecessary animal use, lower development cost, accelerate timelines, and improve human relevance. The difference between the two is not the number of studies. The difference is the quality of the evidentiary logic. The FDA guidance forces sponsors and CROs to answer a harder question: is this evidence package sufficient because it is smaller or because it is better? BBIU structural judgment. This is not simply a reduction of animal testing. It is a redistribution of evidentiary responsibility. That judgment is defensible because FDA's guidance makes streamlined toxicology dependent on pharmacologically relevant species, target binding data, weight of evidence risk assessment, fit-for-purpose NAMs, and early regulatory justification. The main limitation is that the guidance is still draft, limited in scope, and focused primarily on certain oncology, biologics, and conjugated products. Its broader economic and operational consequences will depend on implementation, sponsor behavior, FDA review practice, and the pace at which CROs adapt. What the public version does not cover: this public version preserves the strategic signal while withholding the operational layer. The institutional version expands the analysis into CRO exposure mapping, sponsor decision criteria, Module 4 and Module 2.4 construction logic, modality-specific evidence pathways, market exposure segmentation, and tools for distinguishing disciplined regulatory streamlining from underdeveloped non-clinical strategy. Institutional version availability. Access to the institutional version is available for organizations with a defined strategic, regulatory, industrial, or investment decision context. Requests should be submitted through BBIU's Structural Decision Context channel. When BBIU analysis creates friction, the friction itself is not the issue. The issue is what that friction reveals about structural exposure. This episode presents the public version of BBIU's analysis. The institutional version goes further. It includes the full structural assessment, deeper regulatory and geopolitical mapping, capital risk implications, and the decision-level interpretation designed for investors, executives, and institutions facing high-consequence uncertainty. BBIU's public content is intended to frame the issue. The institutional version is designed to support judgment. To request access or submit a confidential decision context, visit .biopharmabusinessintelligenceunit.com.